Dear colleagues, let's get ready for the session number 7. It will be chaired by me, Maija Dambrova, and colleague Pablo Garcia Roves. We will have presentations on topics related to perspectives of bioenergetics in health and disease, and also we'll start to listen to presentations in the topic of mitochondrial pharmacology, and it's my pleasure to introduce the first speaker, Moritz Eggelbusch. So, floor is yours, and time starts hopefully now when the when it will start. So, go on.
Thank you very much for the introduction. So, physical inactivity, for instance in the form of bed rest, is known to contribute to skeletal muscle insulin resistance. And since our muscles are the major site where we burn fats and carbohydrates for fuel, if they fail to do so properly, this ultimately can lead to the development of diseases such as type 2 diabetes. Now, metabolic inflexibility, the failure to efficiently adapt substrate oxidation depending on demand and supply, is known to be an underlying factor of these inactivity-induced metabolic alterations in the muscle. However, the question remains whether such a mismatch between nutrient uptake and utilization rates actually precedes reductions in mitochondrial respiration. We investigated this in context of a bed rest study conducted by NASA and ESA at the German Aerospace Center, Cologne, Germany. And in this study, 24 healthy individuals underwent 60 days of bed rest. And blood and vasus lateralis muscle biopsies were taken at 3 distinct time points: before the bed rest started, 6 days into the bed rest as a measure for short-term inactivity, and then again after 55 days of bed rest as long-term physical inactivity. 6 days into the bed rest, participants showed initial signs of insulin insensitivity, since the HOMA-ER score, the homeostatic model assessment for insulin resistance, which is derived from fasted blood glucose and insulin levels, serving as an estimate for insulin resistance, the HOMA-ER score was significantly increased already after 6 days of bed rest. Moreover, by performing immunofluorescence microscopy, we observed translocation of the glucose transporter type 4 towards the muscle cell membrane, being essential for glucose uptake. This GLUT4 translocation was significantly decreased, again suggesting insulin resistance. Moreover, we observed an increase in both glycogen and lipid droplets in electron microscopy images of these bedridden muscles, taken together being indicative of a nutrient overload in the muscle. And lastly, metabolomics revealed an upregulation of pathways related to the glycolysis. And to elaborate on these metabolomics data a bit more, what we did here, we investigated more than 100 different metabolites, and we performed an enrichment pathway analysis to see which metabolic pathways would actually be altered as a response to the bed rest. And as I was just mentioning, after short-term inactivity, pathways related to the glycolysis were significantly upregulated, whereas after 55 days of bed rest, we saw a decrease in fatty acid oxidation. And overall, these changes in metabolism really appear to manifest throughout the bed rest period, suggestive of continuous alterations in the skeletal muscle. And to follow up on this, we performed high-resolution respirometry using the Oxygraph 2K. And whereas short-term bed rest did not elicit any significant alterations in mitochondrial respiration, long-term bed rest resulted in significantly reduced NADH and succinate-linked respiration, and furthermore, oxidative phosphorylation capacity was significantly impaired, overall suggesting a reduction in mitochondrial respiration as a response to 55 days of bed rest. And interestingly, these reductions in mitochondrial respiration were in line with the decrease in the protein content of the individual complexes of the electron transport system, as well as ATP synthase, indicated by immunoblotting. And likely, this was driven by a decrease in mitochondrial density, which again we assessed in electron microscopy images of these bedridden muscles, and which you can see depicted here as the mitochondrial surface area. So to summarize and to conclude, bad rest resulted in a shift away from fatty acid oxidation towards glycolysis, from which we can infer metabolic inflexibility. And the inactivity-induced nutrient overload and insulin resistance indeed preceded reductions in mitochondrial respiration. And therefore, our findings suggest a nutritional oversupply to actually be a major determinant for inactivity-associated skeletal muscle alterations that can be targeted by novel interventions. And with this being said, I'd like to thank all my contributors, everyone who contributed to this work. Thanks to you for your attention. And I also brought a poster with me, so should you have any follow-up questions, I'm happy to discuss those with you later on. Thank you.
Perfect timing. It's no time for questions, but definitely I'm sure you will get many. And now we continue with the second speaker for the session. That is Brian. I think you are there. You are not now? Yeah. It's you. On the program, at least. We'll see the presentation. No, it's him, I think. Yeah, now it's Richie Goulding. Yeah. And the floor is yours for the next presentation. Your title is Aberrant Mitochondrial Ultrastructure, despite no difference in mitochondrial respiration in type 1 diabetes. So,
thanks for the introduction. So with the discovery of insulin in the last century and advances in modern medicine, people with type 1 diabetes are now able to live into the 7th or 8th decade of life. But a relatively underappreciated facet of this condition is its impact on skeletal muscle. And that's really unfortunate because, of course, skeletal muscles are an extremely important organ for whole body glucose homeostasis. A number of groups now have reported the presence of mitochondrial dysfunction in individuals with type 1 diabetes that are young and meeting physical activity requirements. The fact that they're already active suggests that somehow their muscles are not responding to exercise training in the way that healthy people do. So it remains to be seen whether the mitochondrial impairment seen in people with type 1 diabetes can be ameliorated by a period of exercise training. So the aim of this study was to compare mitochondrial function and ultrastructure, as well as exercise capacity, between individuals with type 1 diabetes and healthy controls, and to see whether 4 weeks of endurance training could alleviate mitochondrial impairments in both of these populations. So data collection is still ongoing, but so far we've recruited 17 otherwise healthy subjects with type 1 diabetes and 15 healthy controls, which are matched for age, sex, and physical activity levels. These subjects undergo cardiopulmonary exercise testing to assess their exercise capacity, and we also obtain skeletal muscle biopsies with which we perform high-resolution respirometry to determine mitochondrial function, and we use transmission electron microscopy to determine mitochondrial ultrastructure. A subset of both of these groups undergo a 4-week period of endurance exercise training, so we can see if exercise will improve mitochondrial function in these individuals. We found no differences in maximal oxygen uptake between the groups when assessed at baseline. Also, the peak power attained on an incremental exercise test did not differ between the groups, so we can infer that whole body exercise capacity didn't differ between these groups. As expected from the in vivo findings, we didn't see any differences in leak respiration, maximal oxidative phosphorylation capacity, and electron transport capacity. So at baseline, it appears that there's no differences in exercise capacity or mitochondrial function between these groups. There is an important caveat, however, which is that this cohort of individuals with diabetes were relatively better controlled than previous work showing impairments in mitochondrial function. So HbA1c values in our group were around 6.5, and previous work showing impairments has been in individuals which are above about 7.5. So this suggests that if your diabetes is relatively well controlled, you can offset the impairments in exercise capacity and mitochondrial function associated with this disease. So despite no apparent differences in mitochondrial respiration, we next wanted to look, or wanted to see if these mitochondria look normal. So we assessed their ultrastructure using electron microscopy. Here we have a mitochondrial profile of a young individual with a relatively high VO2 max. And you can see, as you'd expect, the crista are nicely organized and really densely packed within the mitochondrion. And this is expected for this individual to be able to attain the ATP synthesis rates required of a VO2 max value such as that. But we do see clear differences between the controls and the patient. So if we look at a patient with very similar VO2 max value, we can see that the mitochondria are rather fragmented, and we can also see that the crista are much less densely packed and relatively disorganized. So this suggests that maintaining a high aerobic capacity isn't sufficient to offset the deleterious effects of the disease on mitochondrial ultrastructure. Finally, we looked at the responses to exercise training. So we saw that 4 weeks of endurance exercise training increased maximal oxygen uptake in control subjects, but it didn't improve maximal oxygen uptake in our type 1 diabetes cohort. So we saw a blunted training response relative to healthy controls in this population. Despite this, we saw no changes in mitochondrial function in either group following the intervention. So the improvements in the controls must have been brought about by adaptations in other systems. So these findings suggest, again, a blunted training response in individuals with type 1 diabetes. So just to conclude, our data suggests that well-controlled type 1 diabetes doesn't appear to result in impairments in exercise capacity or mitochondrial respiration. We do see, however, lower crystal density in individuals with type 1 diabetes. There's a clear discrepancy between our functional data and our imaging results, and we're currently working hard to try and resolve the reasons for that discrepancy, but we can only speculate at present. Finally, 4 weeks endurance training improves exercise capacity in healthy controls, but doesn't seem to do the same in subjects with diabetes. With that, I'd like to thank my collaborators, whose names you can see here, and I've also got a poster next door, so if you fancy asking some questions, I'm happy to have a chat afterwards. So thanks for your attention.
Thank you. So a short presentation, questions later. The next short presentation will be from Beatrice Chabi. The talk will be about coordination between mitochondrial associated membranes, mitochondrial function, and protein synthesis in exercise and myopathic muscles. The floor is yours, and time starts now.
Thank you for the introduction. So today we're going to talk about mitochondrial function and protein synthesis. As you may know, in skeletal muscle, mitochondria are a key organelle to provide ATP for skeletal muscle contraction. But in skeletal muscle, mitochondria is also involved in providing ATP for the protein synthesis, a process that is highly demanding in energy. And we show in the lab that these processes and mitochondrial function are related through specific interaction between mitochondria and sarcoplasmic reticulum, and those interactions are called MAM, mitochondrial associated membrane. And we show that in stress conditions, those interactions is going to decrease to preserve the energy in skeletal muscle for other processes. And today, I'm going to show you some data showing how mitochondrial respiration and protein synthesis are talking and in two specific conditions. So the first one is the condition of acute exercise, and we compared a sedentary group and an acute exercise group, and a third group, acute exercise, followed by 3 hours of recovery period. And we look at the crosstalk between mitochondrial respiration and protein synthesis in two types of muscle, soleus and EDL. The second condition is the DMD model, the MDX mice, where we look at this crosstalk in 3 muscles, soleus, EDL, and diaphragm. To do so, we use the respiration protocol Suite 8 and on muscle homogenates. And to measure protein synthesis, we use the sunset technique based on the pyromycin incorporation in protein. So our first results in soleus show that oxphos respiration and ET capacity are decreased in the acute exercise group, and then it goes back to the basal level after the recovery period in soleus. When we look at the protein synthesis, it's decreased in the acute group, and it's still low in the recovery period. When we look at the EDL muscle, it's kind of the opposite. We have a decrease in the acute and recovery group, even if it's not reaching significance. And for the protein synthesis, it decreases again with the acute exercise and go back to normal in the recovery period. So basically, mitochondrial respiration and protein synthesis are differentially altered in the recovery period, and the way it's going to move depends on the muscle typology. When we look at the DMD model, we see a significant decrease in the diaphragm in the MDX mice, in the soleus, but not in the EDL. And when we look at the muscle protein synthesis, it's really increased in the diaphragm, and it seems to be increased as well in soleus and EDL, but it didn't reach as well significance. So once again, it seems that mitochondrial respiration and protein synthesis are differentially altered in this disease model. So far from this data, it seems that this crosstalk between respiration and protein synthesis is not so simple and is not positively correlated. We saw that in acute exercise, it's specifically in the recovery period, and it seems to depend on the skeletal muscle typology. In the DMD model, it's highly there is an inverse correlation, and specifically in the diaphragm muscle. So our work here has to be continued, especially we want to go on with the signaling pathway regulating protein synthesis, mitochondrial activity, looking at for EBP1 inhibition, AMPK activation. We also want to figure out what is the ATP source for the protein synthesis. Mitochondria, glycolysis, how those two pathways contribute to protein synthesis. And of course, we want to analyze mimes, as it was in my title, but today there is no results right now. With that, I want to thank all my colleagues from the DMEM unit, my students, Marie and Leah, who did a good job on this work, and my fellow colleague who is leading this project with me. Thank you for your attention.
Thank you, Beatrice. And now, Brian, yes, it's your turn. Then you go. Oh, careful. Next speaker is Brian Irving, and he's going to talk about impact of nitric oxide promoters on mitochondrial bioenergetics in a murine model of Alzheimer's disease. So, floor is yours.
Yep. Thank you for the invitation for presenting some of our recent data and for the introduction here. Oops. There we go. Yeah, so we're going to talk a little bit about Alzheimer's disease. Again, it's one of the leading causes of cognitive impairment in older adults. And again, it is highly genetically driven, but there is some lifestyle factors. If you look at obesity and diabetes, it increases the risk by 50% for Alzheimer's disease. In addition, obesity-related reductions in cerebral vascular blood flow is also associated with accumulation of beta amyloid, which is really one of the hallmarks of Alzheimer's disease. And the accumulation of beta amyloid, at least in the animal models, has kind of showed the induce some mitochondrial dysfunction. But collectively, there's kind of this reduction in nitric oxide bioavailability in the brain that does relate to both reduced blood flow in the brain as well as reduced mitochondrial function. And so our overall collectively, these changes contribute to the decline in cognitive function. But overall, our hypothesis is that if we restore nitric oxide bioavailability, can we improve vascular function, which we're not presenting today, but also the mitochondrial bioenergetics. So we tested this hypothesis in a transgenic animal model here that has transgenic for the amyloid precursor protein and preslin-1. Which also has been shown by other groups to have a mitochondrial defect. Now here's our overall protocol. We take the transgenic mice, the littermate controls. The littermate controls were randomized to either normal chow or high-fat feeding starting at 10 days for 6 months. And then the transgenic mice was 45% high-fat diet, normal chow. And then for the nitric oxide kind of donors, we had sodium nitrite in one group, and they're drinking that in the water at 100 milligrams per liter, or L-citrulline, which can be converted to arginine in the kidney. But you can look at just the body mass here over the 6 months. All the groups that had high-fat feeding were gaining body weight clearly compared to the normal chow group, as well as body fat. And then at the end of the 6 months, after euthanizing the mice, we took out the hypothalamus, we homogenized the hypothalamus, and then we put the homogenate in our Ouroboros, and we run a modified soup protocol here in Miro-5, but we also add alpha-chaconin to permeabilize the synaptosomes. And we measured leak, respiration, oxphos, complex N-linked oxphos, NS-linked oxphos, ETS, etc. And here's kind of some of the take-home results early on. If you look at the wild type, there's really no difference between pre and post diet, whether it was chow or high-fat fed diet, but there was an effect of the transgenic Alzheimer's disease phenotype, and particularly in the high-fat group here. Now, when we looked at those that were on either sodium nitrite in their water or the L-citrulline dose in their water, it actually tended to recover oxphos capacity for N-linked substrates as well as NS-linked substrates, and then we kind of uncouple them to look at ETS capacity, and that defect is still there. What's interesting is that if we add rotenone, then and look specifically at complex 2, that defect goes largely away. It's not completely gone. But then we went on and added glycerol 3-phosphates, and again, we see that defect come back. And so kind of our take-home message early on, again, this is fairly preliminary data, we need to do some mechanistic follow-up studies, is that the transgenic APS1 mice here have lower oxphos capacity, and other groups have kind of shown that, as well as ETS capacity. And it's really particularly for the N-linked substrates as well as the glycerol 3-phosphate substrates. And both the sodium nitrite, which is a non-enzymatic way of increasing nitric oxide, as well as L-citrulline, which requires enzymatic conversion to arginine to increase nitric oxide bioavailability in the drinking water, can at least attenuate some of those reductions. Thus, it might be a physiologically relevant nutrient pathway, the target for improving Alzheimer's disease-related mitochondrial pathology. Thank you. And I'd just like to, again, thank my collaborators, Tim Allerton and Paul Soto, which are kind of driving the animal part of the study. I just do the bioenergetics, and then our students that kind of worked on this project as well. Thanks.
Thank you very much. So the next talk will be 10 plus 5 minutes, if I am correct, and Johnny Stiban will be presenting a mitochondrial complex 1 deficiency disorder related to protein, nucleotide binding protein-like. So floor is yours, and time starts.
Thank you. Thank you. I would like to thank the organizers for allowing me to present my work here. And I will be discussing mitochondrial disease, mitochondrial complex 1 deficiency disorder, related to a protein, another protein, NUBPL. We all know complex 1, NADH dehydrogenase, NADH ubiquinone oxidoreductase, is the largest of the electron transport system. And it's made up of 45 protein subunits with very important prosthetic groups, the FMN, and 8 to 10 iron sulfur clusters that are absolutely required for the electron transport capacity of the protein. So these iron-sulfur clusters need to be incorporated into the protein. And here I'm discussing the nucleotide binding protein-like, or NUBPL, initially called iron-sulfur cluster transfer protein, specifically for NADH dehydrogenase. In 2009, it was discovered that this protein, deficiency in this protein, leads to severely dysfunctional mitochondria, and therefore it was absolutely required for the assembly and maturation of complex 1. Yesterday, very nicely, Nick Lane explained to us that iron-sulfur clusters can originate spontaneously. However, in mammals, there are other types of enzymatic reactions that are necessary for the incorporation of iron-sulfur clusters. The mitochondrial iron-sulfur cluster system and the cytosolic iron-sulfur cluster system. Just to make a long story short, there are several proteins that are necessary for the incorporation of iron-sulfur clusters. It starts by cysteine desulfurase that removes the sulfur from the cysteine into the cork ISC complex using electron transfer from NADPH and ferredoxin reductase and ferredoxin. It transfers the electrons with the iron that is imported into the mitochondria, and this, using multiple chaperones, forms the 2-iron 2-sulfur cluster, which can be transported into proteins, or it can be transported outside of the mitochondria for the use in the CIA machinery and in iron homeostasis, or another iron can come in and other electrons coming in using the same system to make the 4-iron 4-sulfur clusters that can be transferred, can be imported into its own proteins, for example, by NUBPL for complex 1, or NFU1 for complex 2, etc. So NUBPL is an extremely important protein for the complex 1 health. And genetic defects in NUBPL lead to a subclass of primary mitochondrial disease. It's called complex 1 deficiency nuclear type 21, with a concomitant increase in complex 2 activity as a compensatory method. In HeLa cells, knockdown of NUBPL leads to mitochondrial remodeling of the respiratory complexes, leads to a loss of cristae membranes and increased lactate production because of over-reliance on fermentation. And of course, there is an absence of proper function of the electron transfer system. The first patients to be discovered with a compound were compound heterozygotes having 2 different mutations in the NUBPL gene. The paternal allele was a missense mutation in the conserved glycine, glycine to arginine, whereas the maternal allele had an exon 7 duplication and a deletion in exons 1 to 4. This first patient had developmental delays, leukodystrophy, and elevated cerebrospinal fluid lactate due to complex 1 deficiency, and other mutations were observed and studied along the lines. So NUBPL, the disease, it leads to a deficiency in complex 1, and it is inherited as an autosomal recessive pattern. It's mapped to chromosome 14q12, and it has multiple neurodegenerative manifestations summarized in this table. Of course, since it is a mitochondrial disease, it affects mostly all tissues, and all tissues will have problems with this in this genetic disease, and multiple outcomes can come out. So this protein has a similar, it's similarly to cytosolic NTPases in the CIA pathway, and like them, it does not have the ability to coordinate iron-sulfur cluster on its own. So it's likely a dimer, because this dimer can incorporate the iron sulfur cluster to transfer it to other proteins. So we actually identified the Drosophila melanogaster homologue of this protein. It was essentially called CG3262 protein, and it shows stark homology to the human NUBPL. We cloned it, we overexpressed it, and we localized it to mitochondria in immunofluorescence and in Western blotting. And we purified this protein as a dimer, irrespective of its iron-sulfur cluster status. So it has a Stokes radius of 37 angstrom, which is estimated molecular mass of 66, corresponding to the theoretical mass of 62 kilodaltons. So it is purified as a dimer. Very interestingly, when we overexpress this NUBPL homologue in Drosophila S2 cells, the results is starkly similar to overexpression of mitochondrial DNA helicase, whereby replication stalling is now removed. So it actually helps in mitochondrial DNA replication. So we also checked the membrane binding. Of course, this is specifically for NADH dehydrogenase, so it has to bind to membranes, and you can see here that using different types of assays, we were able to show that there's a nice binding depending on liposome concentration, and it does not depend on any cardiolipin, any specific lipid content of the liposomes, whereby the other protein, NTD, which is the N-terminal domain of the mitochondrial DNA helicase, shows a strong dependence on membrane binding on cardiolipin content. So both of these proteins bind to the mitochondrial inner membrane. And there has been a paper showing that these two proteins might be interacting. This is very interesting because the Drosophila form of NTD has, the Drosophila form of the helicase has an iron-sulfur cluster, and it is possible that the NUBPL transfers the cluster to the helicase to induce the activity. So basically what I wanted to show you is that NUBPL is a devastating genetic disease characterized by problems in complex 1 and OXPHOS, and it may have implications on mitochondrial DNA replication, and the studies are actually ongoing in my lab. I want to also point that mitochondrial DNA helicase contains iron-sulfur cluster in the N-terminal domain, and this is required for the stability of the protein. And the NTD binds to cardiolipin-rich liposomes, and NUBPL bind to membranes with no lipid specificity, and it could be a binding partner between NUBPL and the helicase to transfer iron-sulfur cluster to enhance DNA replication. How is this related to the disease? How it is related to the human mitochondrial DNA helicase which has no iron-sulfur cluster is currently under investigation in my lab. With that, I would like to acknowledge the great work by a former undergraduate in my lab, Muna Abdrabbo, and the collaboration with Lori Kagoni and Minyan Song at Michigan State University. Thank you very much.
Thank you. The perfect timing. So now we have time for questions. Are there any?
Great talk. I was wondering whether in the Drosophila mutant of NUBPL, you have a compensatory effect of dysfunction of complex 1 by bisphosphate dehydrogenase.
This is something that is currently under investigation. I don't have the right answer right now, but it is something that we are actually actively looking at. Thank you.
More questions?
It's not on.
From the epidemiological side, so iron deficiency is a very strong contributor to the global burden of disease, and it, of course, affects women more than men. And iron is also important for the synthesis of neurotransmitters like dopamine. And it is also relevant, so in Alzheimer's, for example, there is accumulation of iron in the brain causing neuroinflammation. Do you also see in your experiments any signs of inflammatory processes, and is there any kind of interplay?
We actually looked at in cells, basically. We did not move to an organism level, so I don't have an answer to your question, but I think this is something worth looking into.
Thank you. More questions from the audience? If no,
I can. Or you can. No,
okay.
No, go ahead, please.
I was going to ask also about additional models to Drosophila, because it's clearly very important clinically. So what do you see? Do you see any models further in maybe rodents or?
Well, mammals, for the mitochondrial DNA helicase in mammals, basically in rodents or in humans, we do not have iron sulfur clusters. So we are actually looking into that at the moment right now. We just want to see if there is any interaction between these two proteins, and if there is an interaction, what does it do to the protein? Does the overexpression help with the replication stalling or not? Because it's clearly shown in the Drosophila, but the Drosophila requires the iron sulfur cluster in the mitochondrial DNA helicase. We don't. Does it do any difference or not? We're still looking into that.
Any other questions? No. Then I will try. As you know, there are many mutations in complex 1. No? So then in the context of this perspective, this mutation that you are describing, how it relates in the assembling of the complex 1? Or in the functionality you already saw with the OSFO system, but it affects more to the M module or the assembling of the complex 1 and the proper functionality, or what is the main feature that difference from the other mutations?
So basically, it's this mutation is not in complex 1. So this is a mutation, it's in a different protein that is required for the assembly of complex 1. So basically, if this mutation is present, or these mutations, because there are multiple mutations in this protein, the complex 1 is actually formed. It is deformed. It's formed, but not in a correct way. So the iron sulfur clusters in complex 1 are needed for functionality of the enzyme, but they're also needed for structure of the enzyme. So the membrane arm, the matrix arm is a little bit deviating from the normal here.
Thank you. Any other question? Adam, last one, quick. Yeah.
Just a quick follow-up. Have you done any kind of super complex analysis on these to see? I'm curious.
We did not, but other groups have started with this work by looking at super complexes. But this is something that is extremely important to look at and see what the whole thing looks like. Thank you. Thank you.
Okay. If there's no further question, we close. Please. Thank you very much. Yes, it's you, Alexander. Now we change again for a 5-minute talk, if I'm correct. And our next speaker is Alexander. I will see properly. Karabatsiakis.
Thanks a lot.
And yes. the title of his presentation will be Effects of 5 Movement, Desynthesization and Processing of Psychotherapy on Mitochondrial Bioenergetics in Immune Cells from Patients with Post-Traumatic Stress Disorder, a pilot study. Floor is yours.
Thank you very much. So first of all, I would also like to thank the organizers for this very kind event. Today I would like to talk about a translational approach where we try to address changes in mitochondrial bioenergetics in the terms of psychotherapy. And this is actually motivated by the fact that we reported and replicated findings of altered mitochondrial physiology in intact PBMCs collected from patients with depression. And interestingly, the impairment in mitochondrial functioning also correlated with the depressive symptom severity measured with self-reports in terms of questionnaires and also by self-ratings made by the clinicians. But the question is, what is the precise nature of this observation? So would we expect a reversibility due to the correlative nature between mitochondrial activity and depressive symptom severity? So, or is it a kind of stress-driven aging effect that we would expect to characterize in these cells? And of course, to investigate this, we need longitudinal studies with interventional approaches. And after we did this study, we got in contact with colleagues in Ulm, so all the work that I'm showing today was done at the University Clinic in Ulm, and they were conducting a study using a relatively new psychotherapy treatment called EMDR, which is actually based on the confrontative element of a traumatized individual with the support of contralateral eye stimulation, which is supposed to stimulate the bilateral communication in the brain across the corpus callosum, which is considered to be impaired in contralateral cross-communication of the brain. So due to the lack of time, I don't have the possibility to give details about EMDR, but it is an approved and evidence-based psychotherapy intervention. And now the question would be, would we expect to find effects? And EMDR has become quite popular because, for example, Prince Harry said that he used EMDR to overcome the traumatic experiences that he suffered from after the loss of his mother. And when we checked the literature for possible biological correlates of EMDR treatment, we saw that there was, for example, an activation reported of the parasympathetic nervous system, and we had cardiovascular responses and also changes on the level of carbon dioxide and oxygen consumption and oxygen distribution in blood. But actually there was a lack of empirical evidence on the level of mitochondrial respiration. So then we got in contact with a psychotherapist, my colleague Visal Tumani, who is doing this EMDR treatments at the University Clinic in Ulm in the consortium of the EDEN project. EDEN stands for European Depression EMDR Network, and we were able to include 3 patients in total, and 1 patient accepted to provide on a longitudinal basis blood samples, and 2 other subjects provided 1 blood sample before EMDR treatment, and then we isolated PBMCs, we cryopreserved the cells, we did the measurements, and then we compared the data that we received from a cohort of non-mentally affected control subjects, as well as a cohort of acutely depressed patients, and to first get a first idea of what we see on the level of mitochondrial physiology. And what we saw was that in comparison to the control group, for the depressed individuals, we saw a significant decline in mitochondrial functioning. But in addition, in these severely traumatized individuals with a comorbid depression, there was an additional impairment in mitochondrial physiology, and overall the profile reminded us a little bit to somewhat a kind of deflated balloon. So actually there is no clear profile, everything looks relatively low, or relatively low expressed on the level of oxygen consumption. And then we additionally, to better understand our measurements, we also assessed the mitochondrial density using the citrate synthase activity by spectrophotometry after respiration. And here we saw that in this EDEN cohort, there was a significantly stronger mitochondrial density in these PBMCs. And then we investigated the sample from this 1 subject that agreed to provide longitudinal blood samples. And what you can see here is that there was a significant decline in the severity of depressive symptoms over time. And what we then did was we again compared the values from this 1 individual to the controls. And what you can see here are the pre-post measurements of the control donors. And now you see that there, in some certain parameters in mitochondrial respiration, there was a significant increase. And here you can directly compare the levels. So for example, here for the uncoupling state, you can see that the levels really went up and reached the level comparable to those of the normal controls. And we also found this for ATP turnover-related respiration as well as spare respiratory capacity. Yes, I'm pushing it a little bit. And again, there was always an increase detectable, and the values always reached the significance level back towards normal controls. And compared to the quantitative evaluation, we also measured the qualitative values. And as you can see here on the graphical representation, everything went back to normal, as well as did the mitochondrial mass, which is also very good. And overall, I would like to say that this is the first study to investigate psychotherapy treatment effects on the level of mitochondrial respiration. And this is pretty promising, and therefore we would like at the University of Innsbruck to get in contact with the Austrian EMDR network to create a large study to better characterize psychotherapy treatment effects on the level of mitochondrial respiration. And I would like to thank everyone who was involved. And unfortunately, Karin de Punda, my colleague since 2021, is sick and she cannot be here today. I'm having her poster outside, and I'm also very happy to discuss her contribution with you. And thank you a lot. Thanks a lot for your attention.
Thank you very much. And I think you will miss the very nice present for being on time. But
yeah, it's okay.
You can talk to Verena.
Okay, thank you.
We move forward now.
Yeah, thank you. So the next presentation, which will be again 10 plus 5 minutes, Werner Koopman, the decile TPP, mitochondrial targeting moiety, lowers electron transport system super complex levels in primary human skin fibroblasts.
Thank you for the nice introduction and for Erich and his team to invite me. So today I will discuss with you some recent study we published on mitochondrial targeting. So there are many ways to target or to get molecules into mitochondria, which you can see here. There are many more. We focused in this study on the triphenylphosphonium strategy, which is used in about 1,000 papers if you look in PubMed. So it's a widely used strategy to attach cargo here, ranging from fluorescent molecules to antioxidants to destructing molecules. They are connected here and then, because of the positive charge, end up in the mitochondrion. So why did we do this study? And it's a little bit of a pity since there are many colors here, which I cannot see very well. I hope you can. So in the past, we have done a study with Trolox, which is a vitamin E antioxidant. And this antioxidant improved the levels of mitochondrial complex 1, so fully assembled complex 1 in patient fibroblasts with isolated complex 1 deficiency. So I'm talking here on nuclear DNA-encoded mutations, which are associated with a very severe phenotype, probably lethal or mostly lethal before the year 5 of age. So we saw positive effects with the Trolox, so we reasoned, okay, let's target this Trolox to mitochondria. So here you recognize the decyl TPP group, so 10 carbons, the TPP here, and here you recognize the Trolox molecule. We have control molecules here, TPMP being only this part, decyl TPP being the targeting sequence without the cargo, and we also, for good luck, for good reasons, put in mitoQ10 here as well. That's used a lot in this mitochondrial targeting work. So if you look at cell viability, so there are 2 dots here for the 5 different molecules. This is concentrations increasing, the Trolox, mitoE10, Q10, and the decyl and TPMP. We did not see a large effect. So these are chronic incubations, because we also saw in the past with Trolox that we need to do 96 or 72 hours of incubation to see effects at the protein level. So that's why we have relatively long incubations here. So we have a patient cell here, the P5175, and this one carries, it's a typical Lee syndrome fibroblast cell line, carrying a mutation in the NFS4 subunits, one of the core subunits of the structure of complex 1 that you just have seen a couple of talks before. So not much is happening on cell viability. So then we tried to increase the level of fully assembled complex 1 in the patient cell. So what you see here is a blue native. So it's a native gel showing you the levels of fully assembled complex 1 with an antibody, with in-gel activity, and with a complex 2 loading control. So what you see here, this is the control. We have a lower level in the patient. We can increase it with Trolox a little bit. We can increase the activity like we have shown before. But then if we target the Trolox, you do not see an increase but a decrease. So we were highly upset by this since we didn't understand it. So how can targeting of a mitochondrial Trolox get rid of the complex 1? So there's hardly any complex 1 left in these cells anymore. So if we look at the control experiments, we can see that these effects are copied in the patient cell line by mitoQ10 and also by the dicyl TPP. So it's completely gone in the patient cell, whereas you also see some effects of these molecules in the control cell line. So the patient cell is very sensitive to these dicyl TPP containing molecules. Also the activity is gone, and you see for mitoQ, you see some effect on complex 2 as well. And that probably has to do with how mitoQ is replenished again, but we can discuss that if you like after this lecture. Okay, so we can also quantify this, so we did this in multiple experiments. So what you can see here is the level of fully assembled complex 1. In panel B shows you the activity of this complex in control in patients. And to make a long story short, this data shows that the dicyl TPP tail reduces the level and activity of complex 1. So we can phenocopy what we see with the mitoQ and with the mito-E10, also with the dicyl TPP by itself. We do not see that for the TPMP. So it's somewhere in the carbon part of the dicyl TPP. Probably that causes the problems. So the next step is to go at supercomplexes. So as you might know, most of the OXPHAS system, especially complex 1, 3, and 4, are generally present in cells in supercomplexes. So this is a nice example from Leo Sazanov's group showing you what you can do with blue native page. So you can have different molecules that you can detect. So so-called supercomplexes here, consisting of various combinations of electron transport chain complexes. You can have dimers that you can detect for complex 5, complex 3, complex 4, and you can also detect single complexes, depending on the denaturating conditions you use. So we used a similar strategy to see what happens at the supercomplex level with these dicyl TPP compounds. Now, this is a complicated figure, so no worries. I have the quantification on the next slide, which I will use to explain everything. But just to briefly talk you through, here we have a Western blot with anti-complex 1. The different treatments are here. So the left part is control cells. The right part is patient cells. So we can look at supercomplexes here. So complex 1 containing supercomplexes. Complex 1 monomers here, which you hardly can see, a little bit here and a little bit here. We can do the same for complex 3. So these supercomplexes contain both complex 1 and complex 3. That's what we can see from this data. And here we have a not so nice, I have to admit, Western blot for complex 2, where we again see the little effect of mitoQ10 here. So if you quantify this, I think it's more clear. So this is the control cell line. I can use that for explanation. So supercomplexes drop by the mito-E10, the mito-Q10, and the dicyl TPP, not for the TPMP. The complex 1 dimers are released from these complexes. That's what you see here by the 3 treatments. And complex 2 is reduced by mito-Q10. And you see the similar effect in the patient cell, only it's worse. So it appears that the patient cell complex 1 is more sensitive to these treatments, compatible with this complex being less stable because of the mutation in NFS7. That's what we think is the reason for this. Okay, one final experimental slide. So we also looked at some functional parameters. We did many things more, but there's no time to show all of this. So we also can see here, if you look at the lower panel for the routine and maximal oxygen consumption rate, that this is also reduced by the dicyl TPP containing molecules, not by the TPMP, and that it's worse again for the patient cells relative to the control cells. Okay, so what do we see here? What do we think is going on? That's in this scheme. So let's start from the top. We have a dicyl TPP containing molecule here. This red part containing a cargo which has some effects. That's what you want. You want this cargo to affect whatever biology you are studying. But we show here that at least in this cell model, we see effects of the dicyl TPP group by itself. We did some fluidity measurements of the mitochondrial membrane. I did not show that here, but we can show that at relatively high concentrations, I have to admit that here, we get a reduced fluidity of the mitochondrial inner membrane by insertion of these dicyl TPP molecules. What we then get is either a destabilization of the supercomplex or of complex 1 or of both, leading to a reduced complex 1 level. Reduced activity, we measured that. I didn't show you that today. Aberrations in morphology, minor. We also see that as well. We see increased ROS levels. I did not show you that. Also in a concentration-dependent manner. So low concentrations are antioxidant. Also of the dicyl TPP by itself. Higher concentrations are pro-oxidant, as measured with hydroethidium oxidation. So then we get a less negative mitochondrial membrane potential. We measured that as well with TMRM. I didn't show you that today. We get a reduced oxygen consumption rate, and we think that this increased ECAR suggests that you have some kind of adaptation to all this treatment by activation of glycolysis. So the take-home message is never do any dicyl TPP measurement without taking along this control. I think that's also the message of the paper that came out 2 weeks ago on this topic, where you can find more information. Okay, then to acknowledge, so these are all the people involved. So especially I want to thank Michael Murphy and Andrew James from Cambridge, who worked, of course, a lot with MitoQ10 for their insightful discussions. My colleagues who participated to, or contributed to this study, and of course the funding agency. And you for your attention. Thank you.
Thank you. Perfect timing. So with this, we start discussion on mitochondrial pharmacology topics. There are questions?
I have a very nice talk. Thank you. We have been developing mitochondrial targeting compounds by tagging them with triphenylphosphonium, like vitamin succinate, metformin, tamoxifen, and we find them very efficient. And for example, MitoTem is under clinical trial now. But to this, we see with MitoTem, we actually see a lot of alterations in terms of mitochondrial function and destabilization complexes, as you do see with the dicyl TPP. However, we have some data which suggests that if we don't add this cargo, this biological, so we call it biological active component, which is tamoxifen in this case, we actually lose selectivity for cancer cells. Right, so it's just my comment on this.
Mm-hmm. Thank you. Yeah, good to know.
Any more questions? Chico?
Kind of related. So I'll stick with the Q10 since you saw some effects with that one as well. And there are other quinone-containing compounds that have been known to redox cycle with complex 1. I'm thinking of doxorubicin and the anthracyclines and cardiotoxic anthracyclines. Do you think that this, at least the Q10 compound, could be inserting into the inner membrane and redox cycling with complex 1 to generate ROS, to create kind of a ROS damage right at that site for that complex and so forth?
Yeah, good question. So according to the previous analysis with MitoQ10, it's recycled by complex 2. So we think it has to it's the only one who also lowers in this study the complex 2 level. So we think this has something to do with the binding of MitoQ10 to complex 2 to be reactivated again. But that's the only thing I can say on that. Yeah, for now.
Have you done experiments where you test your compounds on the cells after you dissipate the potential?
No, we did not do that. I can say, and that's why maybe that relates a little bit, so this is not a complete story. What we see is, for example, that if you have acute treatments, we don't see anything. So if you do 30 minutes with the molecules, no ROS, no degradation of complex 1. So probably it's a time-dependent effect. And also in other cell models, the effects are much less. So if we look at HeLa cells, if we look at HEC cells, if we look at other, let's say, standard cells, which are cultured under completely different metabolic conditions, we see much smaller effects. So there might also be a link between these effects and the type of cell you are using and the metabolic state of the cell that you are using. Maybe that's not answering your question, but I think it's important for, let's say, for general understanding. So this is not black and white. For fibroblasts, we're pretty sure this is what happens. Yeah.
Thank you. So if there are no more questions, thank you very much for your presentation.
And now we move for our last presentation. It's 5 minutes. And then we'll go for the poster session. And this last presentation will be for Jyotsna Batra. And the title of the presentation will be Non-encapsulated dichloroacetophenone, a regulator of essential mitochondrial enzyme, is a potential inhibitor of prostate cancer cell growth. So, please.
Thank you very much. We had an excellent session this morning on cancer metabolism and different aspects of it. So I thought I will take this opportunity to really explain a little bit how a geneticist like me really got interested into cancer metabolism. So my major interest has been on microRNAs, and I'm sure most of you would know that microRNA binds to the mRNA in a complementary sequence manner, and this is how it regulates several of the genes at the post-transcription level, leading to the halting of the protein synthesis. Now, another important aspect is that if there is a single nucleotide polymorphism, which in short we called as SNPs, can hamper with the binding of a microRNA to the complementary sequence of the mRNA. So we call these single nucleotide polymorphism as mirsnips because they are interacting with the binding site of the microRNAs. So as a geneticist, I was really interested in finding out which mirsnips could be associated with the risk of prostate cancer. So we started looking at the genomic DNA of prostate cancer patients, and in this case, we analyzed 22,000 prostate cancer patient DNA and compared it to similar number of healthy controls. And what we found that there were several mirsnips which were associated with the risk of prostate cancer, several of which have been previously described, and we identified several new ones in the genes which have not been previously described, at least by the genetic studies of prostate cancer. Now then we also looked at the expression difference of these genes in the publicly available data sets. And very interestingly, two of the genes related to mitochondrial metabolism, one of which is MCAT and another one is PDK1, which is pyruvate dehydrogenase kinase, was found to be differentially associated with prostate cancer risk. We then, before really embarking onto working on these genes for additional intervention and their role in the cancer, prostate cancer specifically. And I would also like to add that with prostate cancer genesis, Warburg effect has been always thought in a way that it's not the same way it plays its role like in other cancers. So this was an important aspect for us to first validate if this was a real results or these were just by prediction models or only bioinformatics models. So I will not have time to go into the details of these results, but we could show that microRNAs MIR3916 and 877 were able to regulate PDK1 in allele-specific manner when we did reporter gene assay as well as overexpression of these microRNA into the prostate cancer cell lines. This really made us interested into PDK1, and we started looking a little bit deeper into it. And you can see here that in several of the prostate cancer data sets, and this is at the mRNA expression level, expression of PDK1 was found to be upregulated in these prostate cancer patients, and it was also upregulated in metastasis when you compare it to primary sites, as well as in response to the therapy response and also the refraction to the hormone therapies in the patients who are resistant. We then started looking at the effect of knocking down PDK1 using the siRNA approaches, and we see no effect of its knockdown on prostate cancer cell proliferation. However, prostate cancer cell migration was affected by PDK1 knockdown using siRNA. We also did several additional work using the microRNA as well, and we could also identify one of the microRNA was significantly inhibiting PDK1. However, microRNAs can target several of the genes together, so this was very difficult to identify if this was a specific role of PDK1. We then started looking at several of the pharmacological agents and shortlisted DAP as one of the most promising agent, because as we can see here that in multiple prostate cancer cell lines with different metabolism rate altogether, DAP was highly effective in regulating the proliferation of prostate cancer cells, but not of the normal prostate epithelial cells, or at least at very high IC50 value. We could also see its effect on prostate cancer cell migration, as well as on the colony-forming abilities. We also then checked the metabolism-related parameters and could also show its effect on the glycolytic ability of the cells. We also could show that this effect was actually not at the post-transcription regulation, but at the post-translational regulation by affecting the PDC complex. Next, we were really interested in also making it bioavailable using the nanoparticle approach, and this is where we constructed lactoferrin DAP combined molecules, and you can see that these were further effective against the prostate cancer cells, and also very much effective against the chemo-resistant cell line. So we propose that DAP in the form of a nanoparticle complex can be used as a therapeutic agent for prostate cancer. Just to summarize the results, what I have just stated, but we have also quite interested in microRNA and combination of DAP with other therapies. Thank you very much.
Thank you. With this, close the session, and we move to the poster session that will go a little bit late. I hope plenty of time to enjoy and discuss the posters now. Okay.